Hey guys, Miss Cote here, and this is episode 6 of the cell unit. In the last episode, we looked at the 3 processes of passive transport: diffusion, osmosis, and facilitated diffusion. Remember, in passive transport, molecules move from a region of high concentration to a region of lower concentration, and this does not require any input of energy. This episode will focus on active transport. These are the processes that require the input of energy to get the job done. So let's get started. In the last episode, I talked about how facilitated diffusion requires transport proteins to help move certain types of molecules across a membrane. However, since it's moving molecules down their concentration gradient, it does not require energy. But there are some transport proteins that move molecules in the opposite direction, from areas of low concentration to areas of higher concentration. And since that's not how the molecules really want to go, they need a little extra push, and that push is in the form of energy. When energy is needed for any type of cell transport process, then we call this active transport. The main characteristics for active transport are that molecules are moved against their concentration gradient, from a region of low concentration to a region of higher concentration. Because we're forcing these molecules to go where they really don't want to go, active transport also requires energy. Most of the time, this energy is in the form of ATP, but there are always exceptions to the rule. And last, active transport uses proteins in the cell membrane to get the job done. These proteins are all carrier proteins. There are no channel proteins in active transport. Since these proteins are being powered by energy, a lot of the time you may hear these active transport proteins being referred to as protein pumps. One of the best known pumps that gets talked about in biology is the sodium-potassium pump. Like the name suggests, the sodium-potassium pump is responsible for the movement of sodium and potassium ions against their concentration gradients. In the neuron cell, which is the classic example of these pumps, sodium is higher on the outside of the cell, while potassium is higher on the inside of the cell. So the natural movement of these ions would be that sodium would want to move inside the cell and potassium would want to move out of the cell. And when we get to the nervous system, we'll see that there are conditions where this movement is allowed to happen in order to move a signal from one neuron to another. But under normal conditions, this difference of concentrations on either side of the membrane is what the cells need. So let's go through how this pump maintains the difference. Normally, the pump is open to the inside of the cell. In this shape, the protein has a high affinity or attraction for sodium ions. 3 sodium ions can enter and bind to the 3 binding sites on the left side of the pump. This binding will trigger the 3rd phosphate group on ATP to break off and attach to the protein. This is called phosphorylation, and this basically primes the protein to do work. Phosphorylation will cause the protein to change shape, switching to being open toward the extracellular fluid. The sodium ions are released where they are in a higher concentration, so they're going against their concentration gradient. Now in this position, the protein has a high affinity for potassium ions, and they will enter the protein and bind to the 2 sites that are available to them. This triggers the release of the phosphate group, and without the phosphate, the protein will go back to its original conformation, which is to face the inside of the cell. The protein loses its affinity for potassium, and they're released into the cell against their concentration gradient, and the pump is ready to start the cycle over again. For each turn, 3 sodium ions are being removed for the cell, while 2 potassium ions are being brought in. This leaves the cell with an overall negative charge compared to the outside of the cell, which has more of a positive charge. And this allows the cell to maintain an environment that is different from the external environment. This also happens very fast. This pump can cycle 150 times per second. So here's where we are at this point. Diffusion is for hydrophobic or very small uncharged polar molecules. these molecules can pass right through the cell membrane. If the molecule is charged or hydrophilic, it cannot cross the membrane without assistance. This is where transport proteins come in, which can either be channels or carriers. And notice that in all of these cases, molecules are moving from a high concentration to a lower concentration, and no energy is involved, which is characteristic of passive transport. When there's a transport protein involved, and the molecules are being moved from low to high concentrations, and there's a molecule of ATP in the picture, then active transport is happening. A lot of times, you may see a diagram of a process where there is some sort of cell transport happening, and you may be asked to identify, based on the diagram, what type of cell transport is occurring or what type of molecule is being moved. So you'll need to pick up on the clues that the diagram is giving you. One clue would be to look at the concentration of the molecules on each side of the membrane. Is the movement going high to low or from low to high? Another clue would be if a membrane protein is involved or not. If there's no membrane protein, then it would be just simple diffusion. If there is a protein, then it could be either facilitated diffusion or a protein pump. So if that's the case, the big hint would be if there's a molecule of ATP in the diagram, with the jaggedy arrow pointing to where it will be providing energy. And hopefully once you identify which type of transport is going on, you would be able to give an example of the type of molecule that's being moved. Like the example of the sodium-potassium pump, cells are more negative on the inside compared to the outside. The difference in charges across the membrane creates a voltage, or a membrane potential. And when it comes to how ions diffuse across the membrane, we call this the electrochemical gradient. And the transport proteins that are responsible for this, like the sodium-potassium pump, are called electrogenic pumps, which is the main type of electrogenic pump of animal cells. The main electrogenic pump in other organisms, plants, fungi, and bacteria, is a proton pump, like the one shown here. Remember that protons is another term for hydrogen ions, or H+. Energy is used to move the protons across the membrane against their concentration gradient, and this creates an electrochemical gradient where the outside of the cell is more positive relative to the inside of the cell. Electrogenic pumps, such as the sodium-potassium and the proton pump, can be used to create potential energy that can be used for cellular work. In the next unit, we'll be looking more closely at how proton pumps function in creating energy for photosynthesis and cellular respiration. Another type of transport that requires the use of energy is cotransport. The prefix co means together, and in cotransport, we are coupling the movement of two molecules together. One is moving down its concentration gradient, and that molecule is powering the transport of something else, usually against its concentration gradient. So this is a way that active transport can be done without the need for ATP directly. Let's look at an example of how a proton pump can do this. So here at the bottom, we're looking at a proton pump. We are using active transport to push the protons to the outside of the cell. This creates an electrochemical gradient. The outside of the cell is more positive than the inside of the cell. This also creates potential energy. When we allow these protons to move back to where they want to go, by opening up a channel protein, like the one at the top of the figure, the protons will rush through, naturally moving from an area of high concentration to an area of lower concentration. This moving of the protons is now converting potential energy into kinetic energy, the energy of motion. And this energy that's being given off can be used to actively transport sucrose into the cell. This is active transport, but instead of seeing a molecule of ATP in the top picture, the energy for this part of the process is coming from the movement of the protons. The last section of the chapter involves bulk transport, either the uptake of large molecules or a large quantity of molecules. We're not dealing with transport proteins here, but this does involve the fusion of vesicles with the cell membrane or the creation of vesicles from the cell membrane. And for this to happen, energy is needed, which puts it in the active transport category. If you remember back to the endomembrane system from the cell organelle chapter, any proteins that need to be secreted out of the cell are made on the rough ER, get packaged into vesicles, go to the Golgi for a while, gets packaged into another vesicle, and eventually that vesicle moves to the cell membrane. It fuses and becomes part of the membrane, and the proteins are released from the cell. This process of releasing materials out of the cell is known as exocytosis. Energy is needed to change the shape of the cell membrane. The opposite process is called endocytosis, where the cell is taking in materials by forming vesicles from the cell membrane. However, it's a bit more involved than exocytosis because there are different names depending on what's being brought into the cell. So let's look at some pictures. When a cell brings in food particles, or perhaps a bacterial cell that's not supposed to be in your body, we call this phagocytosis. Phagocytosis literally means cell eating. The material will eventually be digested when the food vacuole fuses with a lysosome. Sometimes the cell brings in droplets of fluid with molecules dissolved in them. This is called pinocytosis, literally cell drinking. A specialized type of pinocytosis that requires binding to a receptor is called receptor-mediated endocytosis. When specific solutes, referred to as ligands, bind to the receptors, they group together and pinch in to form a vesicle, bringing the solutes inside the cell. Your book tells the story about how cholesterol is taken up by the cell using this method. And that finishes the chapter on cell transport. Understand that in order to work properly, the cell needs to bring in nutrients and get rid of wastes. Depending on their structure, some of these materials can easily cross the cell membrane, while others need assistance, or maybe even some type of energy input to get from one side to the other. The processes that you've learned about in this chapter play an important role in the proper functioning of the cell. So as always, if you have any questions, please remember to ask them at the beginning of class. And until the next time I see you, be a good human.